Hello everyone and welcome to our today's lecture dealing with replication and transcription. Before we are going to be talking about those two important processes, I would like to make a quick announcement. Some of you already contacted me and asked a few questions about the lectures I delivered last week, and it's excellent. It is much appreciated. However, there is a much easier way to ask questions. To do so, please do use the dedicated Padlet. So if you go on Blackboard and press on PM1A, then you will see of the left-hand side a link called Exam Preparation Sessions. So in those exam preparation sessions, and many of those are going to be face-to-face, and our first dedicated to the genetics contents is going to be fairly soon, we will address all of the questions on the content of those lectures you asked us so far. So if you would like to ask a question, please follow this link and you will end on our Padlet. Padlet is actually very straightforward and useful tool that we personally really like, and students appreciated last year as well. So most important features of this tool is that all of the questions can be asked in anonymous way. So you don't have to reveal your name, just feel free to post your questions. If you post it, your questions will look like this, and we can basically address them already in the comment section, or use those questions if they're particularly good to basically contribute to teaching of all of you guys in our dedicated sessions. So if you have any questions, please try to avoid contacting us via email, because we are getting a lot of those emails and there is actually quite a chance that our answer might be late. However, if you ask on Padlet, we will be notified instantly, and the member of staff who was delivering the respective session is going to be able to get in touch with you and address those in a very easy and nice way. So if you send us questions or post them on Padlet, please do be as specific as possible, and this is really important. Remember, there are no stupid questions. However, if the question is not specific enough, we are not going to be able to help you. So please try to actually even refer to a specific slide within the lecture, and we are going to be able to help you in a much better and more efficient way. Good. So what did we learn in the last week within the genetics lecture series? So as you might remember, we had a closer look at nucleic acids, specifically at RNA and DNA. Both of those nucleic acids are made up of both of the nucleic acids are made up of nucleotides, because they are condensation polymers of those. And those nucleotides are composed of a pentose sugar, means that this sugar has 5 carbon atoms, a phosphate group attached here, and a nitrogenous base attached on this side. And this is the basic core unit of all nucleic acids. Regarding the sugars within the nucleic acids, there are differences between RNA and DNA. Because in the DNA, the sugar is deoxyribose. In contrast, in RNA, the sugar is ribose. And the main difference here is basically this carbon atom and the functional group attached to it. In deoxyribose, this is an H, and there is a hydroxyl group in the ribose. This structural formula of the sugars is actually quite important if it comes to the naming convention of the direction or polarity of nucleic acids. So this is the 1' carbon atom, followed by 2', 3', 4', and 5'. And as I mentioned before, there are actually very specific carbon atoms within those sugars where different elements of the nucleotides are attached. So if we start counting here with C1 molecule, then we have C2 here, C3 and C4. So we have a phosphate group attached to C4 and base at position number 1. As I already mentioned, we should also remember that all of nucleic acids do have a polarity. So we have the 5' end, and we do have the 3' end, right? So those are things you need to remember. The bases within our nucleic acids are joined by hydrogen bonds that are individually pretty weak, but altogether they can be quite strong. In the DNA molecule, both sister strands run in opposite direction, and this is going to be quite important for today's lecture as well. And within DNA, each base is paired with a specific partner. So in DNA, A is always paired with T and G is always paired with C. In RNA, this is slightly different because A is paired with uracil, instead of T. Good. So what are the main differences between RNA and DNA? In human cells, usually RNA is single-stranded. In contrast, DNA is always double-stranded and forms a structure called double helix, which has been discovered by Watson and Crick. In contrast to DNA, which has one main form in human cells, there are three important types of RNA playing an important role in different processes within the cell. And the first type of those RNAs is messenger RNA. And this type of RNA is going to play an important role during the process of transcription, which we are going to be looking at today as well. Second type of RNA present in our cells are the ribosomal RNAs, short rRNAs, and together with transfer RNAs, those two types of RNAs play important role in protein biosynthesis. And we will look at this process in our lectures next week. So, mRNA is generated during the process of transcription, and transcription is going to be the topic of today's lecture, and it is transcribed from the DNA based on the genetic material in the DNA within the nucleus. Afterwards, RNA is exported from the nucleus and carries the information from the nucleus to the cytoplasm. Within the cytoplasm, mRNA binds to ribosomes containing rRNA, and with the help of tRNA, this leads to production of proteins, starting with short polypeptide chain that become longer and longer until fully mature proteins are generated. Good, this is basically what you need to keep in mind from the lectures last week. So what are the learning outcomes today? First of all, after the end of this section, you should be able to outline the basic steps of DNA replication and know the rules of this process. You should also be able to understand the Mendelssohn-Stahl experiment and be able to explain what it tells us. You need to know what a primer for DNA synthesis is and explain the terms leading strand and lagging strand. Finally, you need to be able to explain how eukaryotes overcome the difficulty of replicating the ends of linear chromosomes. Regarding the transcription, you need to be able to describe the process itself, you need to be aware of its machinery and end products. You should also understand why eukaryotic gen products must be spliced, and you need to know how introns and exons are involved in this process. Last but not least, you should be aware of the major steps of eukaryotic mRNA processing. Good, so I showed you this paper already in the last lecture. This is the paper published by Watson and Crick in the prestigious journal Nature in '53, and already then, whilst describing the structure of DNA as a double helix, they actually recognized something very important, and to cite those guys, they claimed that it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. And indeed, follow-up experiments by other groups have been able to provide additional evidence that this is actually the case, that DNA can indeed be replicated. And it has been also revealed that replication needs to be extremely accurate, because one error in 1 million base pairs leads to 6,400 mistakes every time a cell would divide, which would be catastrophic. This could lead to mutations, resulting in development of different disorders, for example, cancer, if a gene is affected that regulates cell division. Indeed, DNA errors and errors in replication are not only responsible for the development of multiple disorders and conditions, but they are also one of the reasons why we age. DNA damage and gene expression errors are quite common hallmarks of aging. This is why this process is extremely important. In addition, replication also takes place at very, very high speed. In bacteria, like E. coli, DNA replicates itself at the rate of 1000 nucleotides per second, which is really, really rapid. So, what do we need in order to replicate DNA in the cell? First of all, we need a template strand. This is the basis, right? Also, we need raw materials. We need nucleotides to be added to the growing new daughter strand. And obviously, we also do need several enzymes and other proteins assisting with this process. It is quite common sense that cell division requires DNA replication, because if a cell divides, we end up with two identical daughter cells that need identical DNA molecules. And this is actually the job of DNA replication. This happens during mitosis, where DNA needs to be copied in very controlled and identical way. Because DNA is a double helix, there are three possible mechanisms for DNA replication. Again, double helix means that our DNA is composed of two different antiparallel strands. So, replication could be a conservative replication. And by conservative, I mean that each strand would produce identical copy, right? So this strand would generate a copy of itself, and this one, blue strand, would also produce an identical copy. This mode of replication would be a conservative one, where the original DNA molecule would be preserved. In addition, also semi-conservative replication is possible, where daughter DNAs would contain one old and one new strand. Again, those are mixed molecules containing one old strand and one newly produced strand. In theory, there is also a third possible mechanism of DNA replication, namely dispersive replication, where old and new DNA were dispersed in daughter molecules. So each DNA molecule in daughter cells would contain a mixture of new and old DNA molecules. So, how could we prove one of these mechanisms correct and the others wrong? This has been actually performed by Mesenson and Stahl actually quite early on, namely in 1958, just few years after the DNA double helix structure has been revealed by Watson and Crick. And in the experiments those guys have conducted, they actually recognize that all bases present in the DNA actually contain nitrogen molecules. As you can see here, there's quite plenty of them in all of the bases we have looked at before. In normal bases, the type of nitrogen is an epitope called N14. This is the normal type of nitrogen. In the experiments, however, Meselson used different epitope to their advantage, the heavy nitrogen N15. They performed centrifugation, and they realized that you can separate those fairly easy from a mixed suspension. So what they have done, they have cultivated E. coli bacteria and added N50, the heavy nitrogen isotope, to the medium. Therefore, dividing cells have been labeled with this 15 and N isotope. Afterwards, they extracted the DNA from the bacteria and performed centrifugation. And what they observed, that if they perform this with original bacteria cultivated in F15, they obtained one band which was only containing the heavy isotope of the nitrogen. However, in the first generation DOTA molecules, they realized there is actually hybrid DNA containing both the heavy nitrogen isotope N15 and also the normal light isotope N14. So at this stage, they couldn't actually answer this question because all of the three mechanisms would show quite similar result. However, the next second generation DOTA molecules provided very clear answer to this question, because they ended up with one fraction containing DNA which contained only the light nitrogen molecule and also additional band containing hybrid DNA. And this was the proof that ENA replication is clearly semi-conservative. Why? Because if we would assume that the conservative replication mechanism is true, then we would have mainly normal molecules in the second generation and only few heavy molecules. In contrast, in the dispersive replication mechanism, we would have only one hybrid band with N15 and N14 DNA. However, as the answer is semi-conservative, we would expect one light band and also hybrid bands, which was the case in the experiment. And this was a clear proof that the mechanism of DNA replication is semi-conservative. With this, I would like to end up the first session of this 3-lecture video series, and I'll see you in session number 2. Thank you very much for your attention.